Hello, this is Dr. Gray, and we are now going to cover material in Chapter 11, which is talking about another type of cell division called meiosis and its important function and role in sexual reproduction. So sexual reproduction is when there is a combination of specialized cells, which we call gametes, from two different parents, and these come together to form a new individual. What happens is that there's a mixing of chromosomes donated through these gametes from the two parents, and this allows for genetic diversity between and among the offspring. Now, this process of combining gametes is different from what happens in mitosis, in which we have one cell that divides and produces two genetically identical daughter cells, meaning the chromosomes are exactly the same as the ones that were in the original cell. Meiosis is going to produce a different effect. So this is sexual reproduction, where you have gametes coming together to produce a new individual. I'd like you to think about, how does this differ from the asexual reproduction? What does asexual mean? So the process of meiosis, so remember that plants and animals have two sets of chromosomes in their cells, one that they received from each parent. So for example, if you are a human, then you receive 23 chromosomes from your father and 23 chromosomes from your mother. And those come together to match up, so you then have 23 pairs. This is what's called a diploid organism, or 2N, where it means you have two copies of every chromosome. So for humans, the N would equal 23. Now there are reproductive structures that produce either gametes or spores, and these are cells that have a single set of chromosomes. And when you have a single set of chromosomes, we call that haploid, or 1N, one copy of chromosomes. So meiosis is the process in which we can generate those gametes. And so we're going to reduce or decrease the number of chromosomes by 1/2, because in most body cells we have 2N. We want to reduce it in half to produce just a single set of each chromosome. And so remember, in mitosis, which is most of the body cells, we go through the process of dividing the nucleus and creating two cells that have the exact chromosomal makeup. Whereas in meiosis, this only occurs in sex cells, which are also called gametes, sperm and egg. And in this case, we're going to go through two rounds of division, two sets of division, which will result in having either an egg or sperm that have only one set of every chromosome, or haploid. So for the human, this would be 23 chromosomes in each of the egg and the sperm, so that when you put the egg together with a sperm, then you go back to having two copies of every chromosome in the new individual. So these two rounds of division are called meiosis 1 and meiosis 2. And here's a diagram which demonstrates meiosis 1, or interphase leading into meiosis 1, or the first division. In both cases, we need to divide up the nucleus. So in this chart, we see we start in interphase, and they're not showing the gap phases, they're just showing the S phase, because this is where we're going to copy all of the chromosomes. So this is a diploid cell. We're going to make a copy of every chromosome, because we are going to then be dividing up the chromosomes. So we have 46. We'll be doubling that so that we can divide up those chromosomes in the human. We then enter into meiosis only if we are a sex cell, an oocyte or a sperm cell. And we're going to go through the same named phases that we saw with mitosis. Prophase, there's a prometaphase, metaphase, anaphase, telophase, and cytokinesis. So the names don't change, but in meiosis 1, we're going to see a few differences. These are important differences. So we're going to go through each one of those phases. They're all put together here, but let's take a look at them individually. So meiosis 1. So keep in mind that there's a normal interphase that comes before. G1, G2, which are growth phases in which the cell is getting ready for the divisions, as well as they're doing their normal jobs, and the S phase where the chromosomes are copied. Now we move into, as we move into meiosis 1, we see that the S phase is going to make those same sister chromatids, and the sister chromatids are going to stay attached at the centromere. So far we're not really seeing anything different than mitosis. So say here this is chromosome number 1. We're going to make a copy of it, so we have an identical copy to chromosome number 1. And we're going to form the centromere and the kinetochores where the spindle fibers are going to attach. Same thing as mitosis. But when we go into prophase, and this is prophase 1, we see an interesting difference. The first thing is that the two sets of homologous chromosomes are going to pair up. So remember, this is chromosome number 1, say that you got from your mom, and here's the copy of that chromosome that you made in S phase. And this is chromosome number 1 that you got from your dad. And here is the copy, that second blue one is the copy that you made in S phase. These are going to attach themselves to one another. So not just the original to its copy, but the two pairs of homologous chromosomes are going to attach via this synaptonemal complex. So this is going to hold all 4 chromosomes together. In this phase also, there's going to be an important process that we call crossing over, where bits of each chromosome can swap between the each of the chromosomes that are present. Visible structures at the crossover points are called chiasmata. We'll see those in a little bit. And the 4 chromatids are held together by chiasmata, and they're called a tetrad. So this whole thing is called a tetrad. So now let's look at that crossing over. So remember, we had these, we showed them in the previous slide, one on top of the other and held together, and they will do that. But for right now, we've just separated them a bit so you can see better. During this prophase 1, we're going to see that there's going to be an opportunity to swap bits of these chromosomes. Because remember, eventually all 4 are going to end up in separate cells. So in this case, we're going to swap this distal bit of this blue one with this distal bit of this red one. And when the process is complete, you see there's a little bit of the red which has swapped with the blue. And so they contain the same genes, but they might be different versions of that gene. So that eventually, when we break these apart, they'll have a completely solid blue, a blue with a bit of red, a red with a bit of blue, and then a complete red. And so these chromosomes now have a little bit of difference, and this allows for variability in the offspring. Remember, these are chromosomes that are going to end up in offspring. So that happens in prophase 1. Now let's move on to prometaphase 1. This is the early part of metaphase, the first metaphase, and our spindle fibers have formed. They did that in prophase 1, just like in mitosis. And the chromosomes are starting to move around and organize themselves. The homologs are still held together at the chiasmata, so they're still kind of close to one another, and the nuclear membrane has completely broken down towards the end of prophase 1. So we're starting to get the chromosomes in their pairs to line up. Now, if this was metaphase, they would all line up in a straight line, nobody next to each other. But we see in actual metaphase that chromosomes that are the same, this is chromosome 1, this is chromosome 1, are going to line up next to each other, not in a straight line. So the homologous chromosomes are arranged at the cell equator with the kinetochores, that's those little yellow dots in the middle, facing opposite poles. So this kinetochore is facing this way, this kinetochore is facing this way. This is different than mitosis. So the maternal and the paternal chromatids are going to orient randomly, and they're mixed up. So remember we said maybe red was the chromosome you got from your mom and the blue was the one you got from your dad. Well, this is chromosome 1. Let's say this is chromosome number 2. You notice that it is the case that you might have them mixed up. It's not shown in this one, but say in a case it could be that the red one is on this side and the blue one is on this side. So that when they break off and go to the opposite sides, it won't be the case that it's all the blues and all the reds. It will be a mixture of blues and reds. And this allows for even more genetic variation once we eventually get to splitting up all 4 of the chromosomes. And that's really key to making gametes that are going to produce different offspring than their parents, so that although they'll be similar to their parents, they won't be identical. So here we have anaphase 1. So now we're going to see the first division starting. And those microtubules of the spindle, they, remember, will pull chromosomes towards the opposite poles. But you notice here, they are pulling an entire tetrad. These were next to each other, not on top of one another. So we're not breaking the kinetochore apart. We are pulling the entire structure this way. And you notice in this particular one, we do have one red that's mostly maternal being pulled to this side, and one blue that's mostly paternal being pulled to this side. So the chiasmata are broken, but the sister chromatids stay together. They're still attached through their kinetochores. We then move into telophase 1 and cytokinesis. So we're almost at the end of the first meiosis. Those separated chromosomes are at the poles. Here's one little pile of them. Here's the other little pile of them. We've kind of rotated this, and we see that the cell is starting to pinch in, which we saw in mitosis. So we're going to pinch in, and then cytokinesis will be complete when there was a complete separation. All right, and so if that cytokinesis does not occur, then the nuclei won't get formed back together again. All right, so at the end of meiosis 1, we see that, remember, we're going to have a division. We see that it's different in plants versus animals. So in cytokinesis, for animals and fungal cells, they do form a cleavage furrow. They pinch in the middle and you form 2 daughter cells. But in plant cells, we form a cell plate. So we form that cell plate and the cell walls are maintained. So it's going to form a new cell wall right down the division, right down the center, and that's how we're going to make the daughter cells. Okay, so here we have a meiosis animation to take a look at. And you can do that at your own time. But what I'd like to do is move on to talking about meiosis 2. So remember, in meiosis we have two divisions. Now the interesting thing about meiosis 2 is that the process is identical to mitosis. The process is identical to mitosis. So we're going to start prophase 1. There will not be any additional copying of chromosomes because we already did that right from the beginning S phase. So we're just going to go right from cytokinesis of meiosis 1 right into prophase of meiosis 2. And remember, we're going to have our combinations, our sister chromatids attached in homologous chromosomes. Then we'll move through each of those steps that have the same names again as mitosis. So let's look at prophase 2. If the chromosomes decondensed in telophase 1, they'll recondense now. So they may not have gone through the whole process of getting to be unseeable. They may stay in kind of a condensed form and go right back into prophase 2. But if they did decondense, they'll recondense now so we can see them. And the centrosomes, if they were duplicated, then if we already had, if we need to remake new centrosomes, which will form a new spindle, they're going to then migrate to opposite poles. We're going to form a new spindle. So in prometaphase 2, the nuclear envelope is going to go away. Again, just like we would normally see at the end of a prophase. The spindle has formed, and each of these sister chromatids with its kinetochore is going to attach to the microtubules of the spindle fibers. But this time, they're not completely lined up yet. We're going to see they're going to line up in a straight row, just like we would see in normal metaphase of mitosis. So here we're comparing meiosis 1, prometaphase, metaphase, and anaphase, and meiosis 2. So remember, when we started meiosis 1, we didn't line up the chromosomes one on top of one another. We lined them up next to each other so that when they go through anaphase, we're going to pull still a connected pair to each pole. In meiosis 2, we are going to line them up one on top of another, and the spindle fibers are going to attach to opposite kinetochores, so that when we go through anaphase 2, we're going to pull an individual chromosome to each side. So this is what's going to end up in each egg and sperm cell, just a single copy of every chromosome. So here's metaphase 2 and anaphase 2. So these events are going to go forward just like they do in mitosis. We're going to line up our sister chromatids, which are the pairs, in metaphase, but we're going to line them up in a straight line, facing their opposite poles. The spindle fibers will attach to each of the kinetochores, and they're going to pull just one copy of each chromosome and its kinetochore to opposite ends of the cell, towards opposite poles. And by doing that, we're going to end up with just one copy of every chromosome that will go to each pole. Now we could start to go through telophase 2 and cytokinesis. The chromosomes will arrive at opposite ends in telophase. So we have here one chromosome, two chromosomes. This organism only has two chromosomes in this diagram, but we only have one copy of each, because here's one of the copies, here's the other copy. Here's one of the copies, here's the other copy. We're going to make four cells, and they're only going to have one copy of chromosome 1 and one copy of chromosome 2. As we pull them apart, the nuclear envelopes are going to start to reform around each of them, and then we're going to pinch in, Cytokinesis, if this is an animal cell, we're going to pinch in and we're going to form 4 individual cells. And they're going to be haploid cells, having only 1 copy of each chromosome. So now let's go through and compare various components of meiosis to mitosis. So number 1, both processes mean we are dividing the nucleus of eukaryotic cells. So we're not talking about prokaryotes, not talking about bacteria. We're dividing the nucleus in these cells, and that's a similar aspect. Here's a different aspect. Mitosis, we only go through 1 division, creating a diploid cell. Meiosis, we're going to go through 2 divisions, creating haploid cells. Here's another division. Homologous chromosomes pair to form tetrads. Tetra means 4. So we form a clump of 4 copies of each chromosome together in meiosis when we start meiosis. We don't do that in mitosis. We have 2 sets of homologous pairs, and they're separate from one another. In meiosis, we allow this crossing over to happen between sister chromatids, and we allow for a random assortment of the chromosomes in that anaphase 1. So remember, you could have a blue pair and a red pair going to, a blue pair of chromosome 1 and a red pair of chromosome 2 going into the same cell. This allows, both of these allow for genetic variation. Whereas in mitosis, you're producing genetically identical cells at the end of mitosis. You start with 1 cell, the cells that result are genetically identical. You're just making absolute copies. And in meiosis, we reduce the ploidy. In mitosis, we are just copying and continuing to have diploid or 2N cells. But in meiosis, we're going to reduce that. We're no longer going to have diploid, we're going to have haploid, or only 1 copy of every chromosome. And you can imagine this is necessary because we're going to put an egg cell and a sperm cell together, and we want to have the resulting organism have the right number of chromosomes. So that means we need to have only 1 set of every chromosome in those gametes. Okay, so here's a diagram that is again comparing with visuals of each of the steps, meiosis versus mitosis. This is also in your text, and you can take a look at that. So now let's talk a little bit about sexual reproduction and why sexual reproduction can lead to genetic variation. There are different life cycles for sexually reproducing organisms. So organisms that can produce differences in their offspring, they can be in one of these 3 types. There's 3 main categories, and these are for multicellular organisms. So we're talking here about the fungi, the plants, and the animals, because most of the organisms that are in the protist group, even though they're eukaryotes, they're not multicellular. So we have diploid dominant, meaning that these organisms, the cells spend the majority of their life being diploid, or having 2 copies of every chromosome, and that's primarily our animals. We have haploid dominant, so they spend most of their life having their cells having only 1/2 of the normal number of chromosomes. And fungi, like mushrooms, do this. And then we have alternation of generations. And plants do this. They spend half their time as diploid and half as haploid. And we're going to go into a lot more details about this in 2nd semester, but this is just an introduction to this. So our diploid dominant, these are our animals. Animals spend most of their lives, their cells are diploid. And the only time we have haploid cells is when we make gametes, eggs and sperm. Those germ cells, eggs or sperm, are produced by gonads, the testes and the ovaries. And we can use mitosis to make more of the starting cells, but then we're going to use those germ cells to actually go through meiosis and produce gametes. So germ cells are diploid. They're the starting cells, but then they're going to go through meiosis and make gametes, which would be haploid. There is no multicellular haploid life stage. So in animals, you never have a version of an animal which is completely haploid, where all the cells are only half, because remember, it's only the gametes. And fertilization occurs, this is when we make a new individual, fertilization occurs when two gametes, an egg and a sperm, fuse together and restore the diploid state. Because remember, these are haploid. We take two haploid gametes, put them together, we make a diploid. All right, so now let's look at some of these other ones that you might not be quite as familiar with. So haploid dominant. These are fungi, which are in the kingdom fungi, and then some algae, which are actually protists, in which the body of the organism, the actual cell of this organism, is haploid. The cells only have half of the number of chromosomes, and this is the way it spends most of its life. When it goes to reproduce, there are special haploid cells in this haploid body from two individuals, and they join together to make a diploid zygote, which then immediately undergoes mitosis to form 4 haploid spores. So this intermediate, a zygote is diploid. That's when an egg and sperm come together. So it does form a diploid, but it doesn't spend much time at all as a diploid. It immediately forms 4 haploid spores, and then these spores go ahead to form the new individual, which is all haploid. So those spores form the new haploid individual by going through mitosis. So these kinds of organisms, fungi and algae, spend most of their life being haploid. Then there's interesting combination of diploid and haploid where you go back and forth. And so this is seen in some algae, which are protists, but they're seen in all plants. So all plants go through this alternation. So multicellular plants spend part of their life as haploid, as only having half of the number of chromosomes, and they're called gametophytes at that point, and they produce gametes through mitosis. Now remember, animals produce gametes, but they produce it through meiosis. So this is a little bit different. The fertilization happens between gametes. That sounds pretty normal, and we form a diploid zygote. So we have an egg and a sperm within the plants that come together to form a zygote. These zygotes then give rise to a multicellular plant, a diploid version called the sporophyte. So the sporophyte is the diploid version. Now when you go out and look at a tree, that tree is the diploid multicellular plant. That's what you're looking at. It's going to spend its life. Then cells of that sporophyte undergo meiosis to produce haploid spores, which then subsequently develop into more gametophytes. And a gametophyte is actually a tiny little plant which is haploid and spends part of its life being haploid before it makes gametes. So this is where there's that alternation. So this organism will spend part of its life being haploid, and then part of its life being diploid as the sporophyte. So as I said, we will spend lots more time in the second semester going into these more details of these alternations and these different types of life cycles. But for now, just start to get a little bit of the terminology. Thank you.